Namaste, myself Jevin Shivi Saxena, Faculty of Physiotherapy and Diagnostic, and my discipline is Medical Laboratory Technology. So, from Jayoti Vidyapeeth Women's University, Jaipur. So, welcome in digital version 2.0. So, my today topic is electron microscope. Firstly, start my topic is uh, history of electron microscope. So, firstly, in 1897, J.J. Thomson discovered the electron, and in 1927, Louis D. Brodgky identified wavelength for electron. In 1929, E. Ruska have thesis on magnetic lenses, and 1931, Knoll and Ruska bullet the first electron microscope. In 1938, von Voorhis and Ruska bullet the first practical electron microscope. So, electron microscope. The electron microscope use electron beams and magnetic field to produce the images instead of a light waves and glass lenses used in the light microscope. Resolving power of electron microscope is far greater than that of any other compound microscope. This is due to shorter wavelength of electron. The wavelength of electron are about 1 lakh times smaller than the wavelength of visible lights. Okay. Method of electron microscope. The specimen to be observed is prepared as extremely thin, dry film or small skin, and these are then introduced into the instrument at a point between the magnetic condenser and the magnetic objective. The magnified images is viewed on a fluorescent screen through an airtight window. The image can be recorded on a photographic plate by a camera bullet into the instrument. Why do we need electron microscope? Electron microscope are limited by the physics of light to 500x or 1000x magnification and a resolution of zero point 0.2 micrometers. In the early 1930s, there was a scientific desire to see the fine detail of the interior structures of organic cells like nucleus, mitochondria, etc. This required 10,000 eggs plus magnification, which was just not possible using electron microscope. Types. Mainly two types of electron microscope. First is transmission electron microscope. So allow one the study of the inner structure, and scanning electron microscope that used to visualize the surface of objects. So principle of working of transmission electron microscope. Electron process a wave-like character, and electron emit into vacuum from a heated filament with increased accelerating potential will have a small wavelength. Such higher energy electron can penetrating distances of several microns into a solid. If these transmitted electrons could be focused images with much better resolution, and focusing relies on the fact that electrons also behave as negatively charged particles and are therefore deflected by electric or magnetic field. So, what is SAM? The scanning electron microscope use a focused beam of high energy electron to generate a variety of signals. At the surface of solid specimen, the signals that derive from electron sample interactions reveal information about the sample. Principle of SAM. Accelerated electrons in and SAM carry significant amount of kinetic energy, and this energy is dissipated as a variety of signals. produced by electron sample interaction when the incident electrons and declarated in the solid sample. These signals include secondary electrons and produce SAM images. So, structure of SAM. So, share the screen. Uh, uh, this structure SAM. So, first electron gun. So, electron gun consisting of cathode and anode. And second, the condenser lens, two types condenser lens, first condenser lens and second condenser lens. So the work is condenser, so control the amount of electron traveling down and down the column. And third, the objective lens. So the objective lens, this is objective lens. So focus work is focuses the beam into a spot on the sample. Okay. And deflection coil. Deflection coil is this two type of deflection coils. So deflection coils help to deflect the electron beam. And SED, that means secondary electron detector. So attract the secondary electron. Okay. And additional sensor detect, that means back sector electron detector, uh, and X-ray detector. Okay. So SAM working. The electron gun produces an electron beam which is accelerated by the anode. The beam travels through electromagnetic field and lens which focus the beam down toward the sample. A mechanism of deflection coil enabled to guide the beam so that it scan the surface of the sample in a rectangular frame. When the beam touches the surface of the sample, it produces secondary electron, backscared electron, and X-rays. The emit secondary electron is collected by SED and convert it into a signal that is sent to a screen which produce final images. Okay. Advantage and disadvantage of TEM. So advantage firstly, so TEM, that means transmission electron microscope, offers very powerful magnification and resolution. TEM have a wide range of application and can be utilized in a variety of different scientific, educational, and industrial fields. TEM provide information on element and compound structures, and image are highly quality and details. And disadvantages: TEM are large and very expensive, laborious sample preparation, operations, and analysis require special training, and sample are limited to these that are electron transplant. TEM require special housing and maintains images are black and white. Okay. Biological applications of TEM in medicine as a diagnostic tool. So important is renal biopsies, cellular tomography. Tomography refer to imaging by sectioning through the use of any kind of penetrating wave. Information is collected and used to assemble the three-dimensional images of the target, used for obtaining detailed 3D structure and subcellular macromolecular objects. And next, cancer research, so study of tumor cell is ultrastructure, and toxicology to study the impact of environmental pollution by on the different level of biological organization. Okay. Advantages and disadvantages of SAM. So advantage, it gives detailed 3D and topographical imaging, and the versatile information gathered from different detectors. This instrument work very fast, and modern SAM allow for the generation of data in digital form. Most sample require minimal preparations actions, and this advantage of SAM. So SAM are expensive and large, and special training is required to operate a SAM, and the preparation of sample can result is artifacts, and SAM are limited to solid sample, and SAM carry a small risk of radiation exposure associated with the electrons that excite from beneath and the sample surface. Okay. Biological application of SAM. So, virology for investigation of virus structure, cryo-electron microscopy. So, image can be made of the surface of frozen material, and 3D tissue imaging help to know how cells are organized in a 3D network, and their organization determine how cell can interact. And forensics. So SAM reveal the presence of material and evidence that is otherwise undetectable. And SAM render detailed 3D images, so extremely small microorganisms, and anatomical picture of insect, worm, spore, or other organic structure. Difference between SAM and TEM. So TEM, electron beam passes through thin sample. So SAM, electron beam scan over surface the sample, especially TEM, especially prepare thin small sample are supported on TEM grade, and SAM sample can be any thickness, and this mounted on the aluminum stem. And TEM specimen stays halfway down column, and SAM specimen stays is a chamber at the bottom of the column, and TEM image show on fluorescent screen, and TEM image show on TV monitor, and TEM image is a two-dimensional projection of the sample, and SAM image is of the surface of the sample. Okay, so conclusion. Since the invention, electron microscope has been a valuable tool in the development of scientific theory, and it has contributed greatly to biology, medicine, and material science. And this widespread use because they permit observation of material on a nanometer and micrometer scale. And although SAM and TEM are large, expensive pieces of equipment, they remain popular among researchers due to the high resolution and detailed images they produce. So thank you so much. This session is completed, and any query in mentioned comments works. And thank you. Namaste.